I've been looking forward to sharing with you some recent work that I hope provides a different perspective on the origin of cellularity. And those of you who know me know that my lab usually doesn't work on this subject, but rather on evolutionary history at or around the time of the last universal common ancestor, and we know a number of things about that ancestor. One thing we know that's relevant to this talk is that that ancestor was most likely cellular. We think that's the case because it had a signal recognition particle system, which is used for delivering proteins to the cell membrane and either secreting proteins out of the membrane or embedding them within the membrane. I know it says ER lumen here, and that's true for eukaryotes, but in prokaryotes, this system occurs on the cell membrane. One of those proteins that was very likely embedded in the cell membrane of the last universal common ancestor was this ATP synthase motor, which is one of my favorite proteins, and it's responsible for forming ATP from ADP and phosphate by way of a proton motive force that is created by a proton or sometimes sodium gradient that's stabilized by, in the case of prokaryotes, a cell membrane. But when we think about the origin of life, we don't know how it happened. I'm agnostic about how it happened myself because I tend to study things that happened much later. But I hope we can agree that it wasn't the type of cellular organism that we see in modern life today. And in several of the most popular hypotheses for what the simple genetic replicators were that led to the origin of life, they didn't really require cellularity at all. Now, that's not to say that the environment itself didn't provide some of the benefits of cellularity. We've heard a lot about concentration both at this session and at the morning session. And so these simple replicators likely required a rich geochemical environment, whatever that environment may be, that provided them with the kinds of compounds, some source of chemical potential energy, and also mechanisms for concentrating those compounds and making reactions thermodynamically or kinetically favorable. But if the last universal common ancestor was cellular and the origin of life was non-cellular, then that suggests that cellularity arose in between these two stages and maybe evolved because it's an adaptive trait. And if it's an adaptive trait, then there may have been some selective pressure that guided that evolutionary process. And we were interested in understanding what those selective pressures, selection pressures might be. And so we designed what's called a digital life simulation, where we kind of abstract some of the activities of organisms, and which allows us to test hypotheses that we couldn't otherwise test in a laboratory setting or using phylogenetic analysis. And so in our digital life system, there are populations of organisms, and they live in an environment with a shared food stock and some predetermined fixed population capacity. Each organism has a genome that has both metabolic genes and cellularity genes. I'm not going to talk about how the metabolism works because I tried it over and over again and I couldn't get it under 5 minutes, so you can ask me questions about it later. But in general, the metabolism allows organisms to process the food particles and then receive some energy reward for how well they process the food particles. That energy is required to continue to tick through the genome, and every time step in the simulation costs each organism one energy unit. Once the organisms have accumulated an amount of energy that's equivalent to 2 times their initial allotment at the beginning of the simulation, they automatically divide, and that energy is split between the 2 resulting organisms. I do want to talk briefly about the cellularity genes. So the cellularity of an organism is some number between 0 and 1 that's defined by the number of cellularity genes that follows this equation here. And a low level of cellularity has the consequence that the organism is essentially leakier. So a low cellularity organism will be more likely to lose genetic material to the environment, but also receive genetic material that's floating around in the environment. It will also be more likely to lose energy rewards that it got from metabolism, but also receive energy that is floating around in the environment. In addition to the mutations that a cell or an organism might have due to this random horizontal gene transfer that's a consequence of low cellularity, the organisms are also regularly, or their genomes are regularly mutated through insertions of any of these genomic instruction types, deletions of those genomic instructions, or small reconfigurations of the metabolic network that they use to process food particles. And after we had created this digital life simulation, we were kind of looking through the different definable parameter spaces and trying to determine what would be a good area to investigate for selection pressures. And one thing that we noticed was when we made the energy reward from processing food very generous, we would always see an evolution of non-cellularity. And what we thought might have been happening is that organisms were starting off with zero cellularity. It didn't take much metabolism, basically the metabolism of a randomized genome, to get a lot of energy, and then that energy was leaking out into the environment, and it behooved organisms to maintain a low cellularity so that, well, that's not the right way to talk about evolutionary processes. It selected for organisms that had a low level of cellularity because those organisms were able to take advantage of the energy pool in the environment. And so that led us to a more formal hypothesis that in environments where there was a high level of not just food, but the energy that is a reward from processing food, so if we give them freely available energy in the environment, that will actually select for non-cellularity, and it's only when we limit that energy in the environment that it will select for cellularity. So we set up a series of experiments to test this hypothesis. Half of those experiments had simulations where organisms began with 0% cellularity, and we either gave them unlimited food and energy in the environment, or we gave them unlimited food but limited the amount of energy that was in the environment. The other half, so that we could assign a direction of selection here, the other half began with a cellularity value of 87.5%, which is equivalent to 3 cellularity genes, and then again they either had unlimited food and energy in the environment, or unlimited food but limited energy in the environment. And we ran each of these different experiments under 3 different maximum population capacities to see whether population pressure played any role, and then we ran each of those simulations 3 times. So each of these experiments has 9 different simulations, and all of the results of those were pretty similar. So I want to direct your attention to panel B here, in which the organisms begin with a cellularity of 0%, but we limit the amount of energy in the environment, and as our hypothesis would predict, we see an evolution toward cellularity. These are population averages of the cellularity across all of the organisms in the simulation, and the cellularity reaches near 100%. I also want to direct your attention to this panel where the organisms begin with a high level of cellularity, but we give them both unlimited food and unlimited energy, and in every one of these simulations, the cellularity value of the average cellularity of the entire population settles at pretty close to 0% cellularity. So our predictions were borne out by these experiments. At the same time that we were measuring cellularity, we were also measuring how good their metabolism was. And we did that by pausing the simulation and giving each organism 1,000 food particles that they would process, and then we took the average quality of their solution to the food processing. And then we averaged that across the whole population, and we came up with a value of metabolic proficiency. Now, a metabolic proficiency of 0 means no metabolism is happening. A metabolic proficiency of 8, for reasons of how the metabolism works that I didn't discuss in this talk, a metabolic proficiency of 8 means a perfect metabolism, and a randomized genome like the ones that they start with is equivalent to a little under 4. So in every scenario where we limited the amount of energy in the environment, organisms had to process food to get energy. That's panel B and D, and in all of those cases, the metabolic proficiency increased over time. But in every case where they gave them unlimited energy, metabolic proficiency did not increase in the population, so they settled at basically the random value, or the value of a randomized genome. So this shows that, and these results aren't too surprising, but taken together, this shows that when there's energy in the environment, that selection is that the selection within our digital life simulation is selecting for populations of organisms that have both a low level of cellularity and a metabolic proficiency that's equivalent to a randomized genome. When energy is limited in the environment, organisms have to process food metabolically, and so we see metabolic proficiency increase, but we also see cellularity increase across the population. And between these two extremes, we see that these two traits are linked. And so this is just the experiment in which organisms started with 0% cellularity, and we gave them unlimited food, but we limited the amount of energy in the environment. And as cellularity increased, so did metabolic proficiency. So these are the data points for all 9 of the simulations in this experiment. And you see that they're very strongly correlated. These two traits are very strongly correlated with one another as they are evolving. And so we take this as evidence that within the context of our digital life simulation, the cellularity and metabolism are actually co-evolving with one another. And we think this is important for the context of the origin of life, because if you consider the kind of geochemical environment that we'd all like to see as an origin of life setting, one that was very rich, that was providing the early simple replicators with all of the materials and energy that they would need to keep going, it's not just the case that these simple replicators were non-cellular when they first arose, but that this environment would have strongly selected for non-cellularity, and it wasn't until a change occurred in that environment that cellularity and metabolism would have co-evolved. I think for the interest of time, because I'd like to take questions, I'm going to skip some other experiments and just point out that there were several co-authors on this work. Yuta Takagi, who's shown here in my lab circa 2015, also sitting right there. Ziap Nguyen, who is sitting right there or here from my lab this year, and then Tom Wexler, who's a colleague who was at Oberlin College but has since left for Google. Also some funding sources who made this work possible, and thank you for your attention.
The ability to do a metabolism is not the only benefit of a membrane. For example, a membrane protects yourself against molecular parasites, and a membrane helps you to define yourself for evolution, which means I think your conclusion is right that once you need to have a metabolism, you need a membrane, but I would not make the other conclusion that without the necessity for metabolism, you don't need a membrane.
Actually, you're absolutely right, and I skipped over the experiment that demonstrates that, so apologies for that. So we actually turned off, because it's a digital life simulation, we can do that, we turned off the consequences of randomized horizontal gene transfer, and when there's unlimited energy in the environment and organisms don't have to do metabolism, we still see a strong selection for non-cellularity. But when there is limited energy in the environment and they have to do metabolism, with horizontal gene transfer turned off, it doesn't really settle. The cellularity doesn't increase to 100% or near 100%. It actually kind of fluctuates around. And you can see we've extended the simulation out to 10 to the 5th time steps, and it's still fluctuating around. And so we think that in the situation where there's unlimited energy. There's selection for not having a very good metabolism, or there's not selection at all for metabolism. It seems to be the equivalent of a randomized genome, but there's selection for non-cellularity to take advantage of the energy in the environment. But when we limit the energy in the environment, the selection is for having a proficient metabolism, and cellularity, just as you suggested, is actually a mechanism for providing genetic fidelity to the organism.
There's an interesting set of simulations. Now, one of the things that you point out is that there's a selection pressure away from the cellularity. And so my question is, are there systems in modern biology, high energy, lots of food, that you can look to and see? And does it make sense to look at modern systems?
Well, I think viruses don't have to do their own metabolism, and they need to have a little spacesuit to get from cell to cell. But once they're in the cell, they kind of become these non-cellular entities within the cell. And so that might be, I can't think of any others off the top of my head, but that might be a nice analogy to the kind of thing we're seeing here.
All right, thank you, and thank you for that talk, and thank all the speakers today.